our first speaker of this afternoon, which is Elizabeth Tapia. Elizabeth is actually the head of the Bioagroinformatics Division at CIFASIS Research Institute in Rosario, Argentina. She's also professor of the University of Rosario, and she's the actual president of the Argentinian Society of Bioinformatics and Computational Biology. So it's a great pleasure for me to present Elizabeth, and she will talk about today automatic annotation of long non-coding RNA functions. Please, Elizabeth, go ahead.
Yes, I will share my screen. Let me see. So we are ready. So thank you, Georgina, for that kind presentation. It's a great pleasure for me being part of this unique event. I'm really happy to know that we have 4 days ahead to share our experience, to leverage our networking, and to hopefully to boost the careers of the next generation of women in bioinformatics. So my presentation is entitled Automatic Annotation of Low Non-Coding RNAs. And this is one of the my research line of my group at the CIFASIS Institute in Rosario, Argentina. So I will start, I will organize and present this this research from bottom to top. That is, I will start defining what we understand by long non-coding RNAs. So the idea is that in the human genome, with more than 3 million bases, more than 80% of the bases are related to the human transcriptome, and less than 3% of the bases are related to messenger RNA, and that's to proteins. So these are good notices for us, for bioinformatics people, because then there is a huge amount of data to work with. And when we talk about RNA and we extract RNA, we can probably will find this picture where most of the material is ribosomal RNA, and only a fraction is messenger RNA, and only a smaller fraction are involved low non-coding RNA, which are broadly defined as RNA molecules which lack of coding potential, and which sizes are above 200 nucleotides. Although this is a broad definition, it's enough for the bioinformatician side. So what we want with these molecules is to infer their function. That is, we want to annotate them. For this purpose, the first we need to do is to understand how they are represented and where they convey the information about their functionality. So when we observe this problem, we find that the problem is quite different from the problem we observe for proteins, because low non-coding RNA encode their information mainly in the 2-dimensional setting. That is, it's not the composition, but the folding which determines its functionality or its participation in certain biological process, or even its localization. So it's when we observe this picture, we find that are special folding blocks that the RNA molecules mostly use. So if this is the picture we need for performing inference, computational inference, we need to extract this information in a systematic way. So the first thing we do is to start to study this representation as provided by another tools that are well studied in the field of bioinformatics, like those that predict the secondary structure of these biomolecules. And in this case, we find another difficulty is that these tools often provide more than one candidate structure. In this case, we will try to advance and leave aside and only will take the top scoring representation. So when we do this, we observe that this molecule unfolds in this way, and we can here find the one-dimensional representation. Here we can find the fault, but this fault, if we leave aside the pseudonodes, can be right in this form. That is, the first line encodes the composition, and the second line encodes in a simple manner the folding of the molecules. So here we have moved from two-dimensional representation to two different linear representations in one dimension, but we need just one representation. So that's a computational problem that, and to overcome this problem, we introduce what we call a coding trick. That is, the coding trick just tries to fuse, to merge these sources of information using an encoding scheme. In this case, you have the 4 letters of the RNA molecules, and here you have the 3 letters of the Bragg notation, and here are the corresponding symbol we use. And in this, using this table, you can merge these two sequences into just one sequence, which is expressed in a higher order symbolic level. And this is a good notice, because in this way we can use standard methods we already know for machine learning purposes. So this is the first step. And the next problem is, how can we infer the annotation? And it happens that the annotation of these molecules is not simple, in the sense that, in fact, they are the concepts for their annotation, for their definition of the functionality, are actually modeled by a Gene Ontology. That is a control vocabulary with constraints between the concepts. And moreover, these concepts are structured so that the concepts are not independent, but organized in a rather hierarchical way. So this is more complex than inferring a simple difference between independent classes. So we need special tools, computational tools, to overcome this problem. So we formally developed some tools to overcome this problem with when predicting protein function. It's important to remark that in the case of long non-coding RNA, we cannot reuse that is similarity annotation, guilt differentiation, because we already know that the functionality is not encoded in the composition. So the idea is to solve the problem in the machine learning framework in a distributed way. Moreover, there is another constraint is that is in this ontology representation, you have 3 subdomains: cellular component, molecular function, and biological process, that have been treated mostly in isolated ways. And when we have long coding RNAs, we have many relationships that go across the 3 subdomains. So we need an integrated view of the Gene Ontology description of the functionality of long coding RNA that allows a complete inference along the 3 subdomains. That is connected inference, and enriched inference in the sense that includes another relationships beyond the classical specialization. Function is you can introduce part of, regulates occurring, and this gives more power to our description of the functionality. So in fact, we succeed in this idea with protein coding genes, but now we are trying to reuse this with long non-coding RNAs. So how we did it with proteins? Inferring this kind of objects, that is concepts that are related by some constraints like this, is a really computational, it entails a high computational complexity. So the idea is to divide the problem into smaller problems. In this case, you can see here we are thinking about, instead of inferring the complete problem, we are trying to infer about individual nodes. And we will infer individual nodes using a standard support vector machines, for example, but you can use another tools, that doesn't matter. But again, we have another problem, because if we allow these nodes to be support vector machines, we know that since the information is not complete to train the individual learners, you will find error here. And since you need to check the consistency of these constraints, you will check erroneous constraints. So you need to put some interface between the the practical predictions and the ideal prediction. And this is this computational model can be accomplished by means of factor graphs, which are already used in the field of error correcting codes to manage incomplete information. So here you have your core graph, and here you introduce functions which covers the gap between the practical predictions and ideal predictions. And here you have functions that allow to check the consistency between the ideal predictions. With this in mind, we can produce a message passing algorithms so that when you allow these border learners to make a prediction, we put all them to exchange message until we find the stable state here in the green nodes, which will give us the correct inference about the functionality of our biomolecule in the best case. So with this idea, we validated using a small model, Gene Ontology model with 500 nodes. And we use data of long non-coding RNA from the zebrafish database. We obtained more than 7,000 of sequences of already annotated. Only a portion of them, a fraction of them, was annotated by experimental methods. The other were annotated by interaction networks. And so we analyzed these sequences and assigned them to each of the three subdomains: cellular component, molecular function, and biological process, including the constraint that each node, or each individual learner, should have more than 50, at least more than 50 sequences to work with, because otherwise the learner will introduce more noise than a contribution to obtain a stable state in the network. So we did this, and using the standard machine learning approach of 80% for training and 20% for test, we evaluate precision and recall and the combined F score in a hierarchical way. That's the first letter of these parameters. And we find effectively that when we perform inference across the three subdomains, you can see here that the performance is improved by a 5% at least with respect to those in the individual subdomains. So this confirms our intuition that performing inference across the three subdomains improves the quality of the predictions. Part of this work has been recently submitted, so we expect results in the near future. So this is the first part of our work. This is a work in progress, and I would like to continue with a small story about another part of our lab. And the idea is that these four letters are magic. While we attempt to learn how to perform message passing in these factor graphs, we did it so because we were trying to design a small oligos which can resist sequencing noise. And the idea was quite simple. When you design oligos, when the idea that is that you use in sequencing in Illumina sequencing machines, and Illumina sequencing machines are rather perfect. But we imagine that someday Illumina may need more resistant, more resilient barcodes. So at that time, we applied for a grant. It was 2011, and we obtained and we devised a family of barcodes that can be used for Illumina machines. But unfortunately, those barcodes require at least 20 bases, which is much larger than the number of bases that Illumina allowed, which was around 12, no more than 12 or 16 now. So we said, well, we are almost done. In fact, it was not true, because in around 2014, it came one postdoc researcher, and in our first meeting, we realized that maybe these barcodes could be used for the immersion long-read sequencing technology, in this case, PacBio machines. But there was another problem, because in that case, we need to improve our designs to allow for indel errors, because these machines produce a high rate of indel errors. So we continue a couple of years, and by 2016, we succeeded in the first simulations. At that time, we realized that we need to move from the paper to the wet lab, the dry lab to the wet lab. And we did it with the help of another group, which was held by an incredible woman in Brazil, Enrique Hassan. There we produced our minimum viable experiment and realized that we could effectively use these barcodes to produce multiplex long-read sequencing in the Oxford nanopore sequencing machines. And so when we returned, it took us around a year to finish this work with all the bioinformatics we needed to analyze this data with these novel barcodes. And around 2018, we built up a startup with the help of CONICET Institute. So it took us 1 year to find the necessary funding. And when we found, we believed that all was done. And it was not the case, because at the 2019, the country perspective changed. Also, there was no funding at that time. But a few months later, around March 20 of this year, the COVID pandemic came, and some people became interested in our development. Finally, we found the necessary funding, and we start our startup. So now we are working also in this development that is trying to devise more efficient methods to perform multiple sequencing samples in one run of NOx or nanopore sequencing machine. And we have people working in an external lab to prove this concept. And we are now working in the multiplex sequencing of 96 samples of the COVID-19 virus. We build this, we build upon previous result that we obtained in Brazil 3 years ago. And so we are also collaborating with another labs, 5 labs in Rosario City with this project. And I would like to, I'm now finishing, I would like to thank my team, and they are incredible. They really work with passion. They like to collaborate. So all this work is possible because of them. So here are our collaborations around the world. And so I just want to say gracias. And I'm willing to answer any question you may have.
Thank you very much, Elizabeth, for this very interesting talk. And I really appreciate you telling us your research lines at the lab, but also your successful experience as being the founder of a biotechnology company. I think this is very interesting, and especially for women.
Yeah.
I think it was very nice. Thank you. And we have 5 minutes questions, if there are any. Let's see, on the web. Okay, I don't see questions here, but I have a few. The first one is regarding the first part of your presentation, when you show your algorithm, the message passes algorithm. You said that it was this problem had a very computational complexity in terms of computation. And so I want to know the complexity or the time that this algorithm takes.
Yes, the message passing algorithm, in fact, it's a well behaves well because it is used in the telephones. Basically, all the telephones, intelligent telephones we have, they have a chip inside, and inside they have a machine that performs a message passing to correct the errors you receive in your telephone, basically, because you are transmitting information in the presence of noise. So I cannot remember exactly the complexity, but it's logarithmic.
Okay, but I mean, it's for a complete genome, is hours, minutes, or days?
No, in fact, it only, you may use time to train, because you need to train. There are two phases. First, you need to train your individual nodes. That will take the time you need to train any support vector machine. The only difference here is that you have many support vector machines, as many as nodes you have in the factor graph. And then when the additional complexity comes when you need to make the inference, because this algorithm is iterative, that is, it requires some rounds around to exchange messages. No, it's quite, it's really fast, and minutes, seconds in the no more than that to make a simple inference. Remember that we are trying to infer the functionality of a single long non-coding RNA sequence. This sequence may be as large as 5,000 nucleotides, and in this case, maybe it will take minutes, but no more than that. It can scale. It should scale. We are working on that issue.
Excellent. Thank you, Elizabeth. I have here two questions. First one from David. Hello, can you mention what type of algorithms and computational power were needed for this project? Well, similarly to my question, but I think he meant computational power, kind of CPUs, GPUs.
Oh, okay. Yes. No, we are using. In fact, at this stage we are using a personal computer or a laptop. We are not using more than that. But of course, if you need to, but we are only working with 500 nodes. Note that when the next step is to move to the human genome, and in that case, we will need more computational power, because the number of nodes, that is the Go ontology graph, is much larger than the Go ontology graph that use the zebrafish. That is, in that case, we will need to put working more than 1,000 of support vector machines to perform individual inferences, and then after that, an algorithms that put all together and give us a consistent annotation in the factor graph.
Great,
at this stage, only your personal computer.
Okay, great, thank you. The next questions from Adonai Alan. I would like to know about annotation tools, because if there is a high complexity for annotation, do the existing tools use machine learning?
I don't know if I understand well, but the problem with annotation, with the annotation of long non-coding RNA, is that mainly you cannot use the standard BLAST approach, that is similarity approach, for the reason I said at the beginning of the talk. That is, the functionality is encoded at the two-dimensional setting. So you need to do, and there are some former approaches that try to perform alignment in the two-dimensional space, but in fact the complexity is impossible. You cannot do that. So you must resort to another ideas. And so this is a proposal that is mainly machine learning based. Of course, you can use whatever you want at individual nodes. You can use even deep learning. I don't know if it makes sense. We will talk about with this about this issue later. But for the problem with the annotation tools is they're often web servers that you don't know what the web servers do in the backend, and they do not provide information about the validation of the annotations. And with this idea, we are trying to improve over that results. So we do not want to provide a solution that is only useful for one type of organisms or that kind of things. We would like to provide a solution that is horizontal to all organisms that would need the notation of long non-coding RNA. For this reason, we are making efforts to provide a formal modeling. These are formal modeling. Note there is no heuristic here. All is formal. It takes more time, but maybe it will pay in the future. Pay off in the future.
Yes, thank you, Elizabeth. And we have time, so I will make the last question. We have 5 more minutes. Regarding, in the last slide, you said that you are working in some project that has to do with COVID-19, and I'm really curious about what is the project, what do you have to do in this project, please?
Yes, what we have to do, in fact, is when the pandemic started, together with our colleagues in Rosario from the different fields, and one expert in virology, the other in question of clinics, we from bioinformatics, and other people from the wet lab, and incredible, we are all women. The head of all these projects, projects are all women. And also our colleague from Buenos Aires, Sofia La Vista. We all worked together and decided to think about the opportunity to sequence locally in Argentina, because we think that a solution should be available for our countries, an economical solution. That is, when you talk about this nanopore sequencing technology, many people say it's it's nice, it's really nice, it's real time, it is not expensive, because the sequencing machine costs you about $1,000, no more than that. But the problem is that when you deploy a sequencing project with these machines, you need at least $1,000 for the enzymes, and you need many other things. So you have real time, you don't need to send your samples outside your country. The sequencing machine is portable, and of course it has different bioinformatics challenges, but we can deal with them. But the problem is that you cannot use $2,000 per sample, even for COVID-19. And the idea was to perform multiple sequences, to sequence many samples from COVID in parallel. That is, in this way we can divide the cost of sequencing by the number of samples. So we are now working, because we did it in the past with chikungunya, and now we are working with 30 samples, but in this case the technology changed, so we need to test again the feasibility of the project. So we tested 12 samples recently, and we are now moving to 48, and then we will move to 96. So we really here in this small photograph at the bottom left, you observe our guys working in the lab, and really we are finishing the first sequencing runs, and we are analyzing the data, and the idea is to develop a sequencing service in our country that is adapted to our economical resources mainly, and of course to provide the opportunity to perform epidemiological policies of surveillance in Argentina. That is to participate in the world goals about the pandemic in a I don't know how, perhaps in the correct manner, that is with the same resources, with the same opportunities. That's our idea.
Excellent.
I don't know.
Excellent, Elizabeth. Thank you very much. I really enjoy your talk, and this project I think is good, very good. And okay, congratulations, and thank you very much for being part of this event. Thank you.
Thank you, Georgina.